Hello, my name is Jack Boulay, and I'm going to tell you about infectious mononucleosis. So the presentation will take the following format. I'm first going to talk about the history and discovery, then the common causative agent, effects of the disease, mechanism of attack, transmission of the virus, and diagnostics, as well as cures and vaccines, both present ones, whether they exist, and also future research. So mono is most often caused by Epstein-Barr virus, abbreviated EBV. Its genus is lymphocryptovirus, and its species is gamma herpes virus 4. There are 8 human herpes viruses, and there are 2 human gamma herpes viruses. Now, over 90% of cases of mono are caused by EBV, so to understand the disease, it truly is critical that we talk about EBV. And I'd like to tell you a little bit about the history behind its research. So these 3 people, Anthony Epstein, Bert Achong, and Yvonne Barr, discovered EBV in March of 1964. Epstein was the first one to learn about the effects of EBV, of this virus, when he attended a lecture by Dennis Burkitt, who was at the time researching tumors in or near the cranial regions of African children. And Epstein was already particularly interested in how viruses may be linked to tumors. Now, scientists already knew that viruses could cause tumors. They knew this since 1911. And so after the lecture, Epstein and Burkitt started talking, and they started talking about whether or not a virus could be the cause of these tumors. And so Burkitt provided Epstein with a sample of the neoplastic cells that he was himself researching. Just as a side note, these neoplastic cells would later be referred to as endemic Burkitt's lymphoma. And Epstein took this sample back to his own lab, where he, Achong, and Barr researched them, and they eventually looked at them through an electron microscope. And they discovered that there were, in fact, viral particles in these cells. And although they discovered this virus while researching Burkitt's lymphoma, this virus is also it's the primary cause of mono. So what is the virus that they found? It's an enveloped icosahedral specialist virus with linear dsDNA genomic structure. So it's enveloped. You can see the EBV envelope has many glycoproteins on the surface, and these are very important in pathogenesis, which we'll talk about in a little bit. You can see it's icosahedral, and that means that the capsid which surrounds its genetic material is it's a regular polyhedron composed of 20 equilateral faces. You can see in between there are matrix proteins which help to maintain structure. It's a specialist virus, which means that it attacks only certain cells within its host, namely B cells and oropharyngeal cells, and we'll talk in a little bit about how specifically it does that. It has linear dsDNA, which means that it is composed of multiple different nucleic acid molecules. Now, although over 90% of the adult population is infected with EBV, most are asymptomatic. Those who are most likely to experience symptoms or express signs are young adults, people ages 15 to 24. Symptoms include tiredness, aching, sore throat. Signs include swollen lymph nodes, particularly the ones in the neck and in the armpits, especially the neck, because there are about 200 lymph nodes in the neck alone. And more signs are enlarged spleen and liver, fever, and rashes. And these they typically arise after 4 to 6 weeks of infection, and they last for about 2 to 4 weeks, although they may last for many months. It depends. So if it's a specialist virus, what exactly does it attack? As I've said, it attacks the oropharyngeal epithelial cells. This is its initial attack. And these are the cells in these regions in this picture. Now, the oropharyngeal epithelial cells are shed from those areas into the saliva, where it is then transmitted, possibly by coughing, by sneezing even, or by kissing, for which reason it's called the kissing disease. Now, EBV attacks germinal centers also. Germinal centers are found in secondary lymphoid organs, such as the lymph nodes and the spleen. Especially the lymph nodes. As I've said before, there are about 200 in the neck. And of course, if it's the kissing disease, then it's the ones in the neck are the ones that are most readily available. Now, it infects the germinal centers because of the abundance of B cells in there. Now, B cells express CR1 and CR2. CR2 is the one we really care to talk about. CR2 is also called CD21, or Epstein-Barr receptor. In a healthy B cell, what happens is C3b here in this picture undergoes mediated cleavage when it attaches to this CR1. It undergoes mediated cleavage by this factor I protease, and it turns into IC3b, which also undergoes mediated cleavage and turns into C3dG, which then attaches to CR2 and lowers the activation threshold of this B cell. However, in an infected B cell, what happens is, so EBV binds, it can bind to CR2 or CR1. It typically binds to CR2, which is why I put this one in parentheses. It's not as common. EBV glycoproteins, they're ones involved in attachment and entry. The ones involved in attachment are GP350 or GP220. And the numbers there just indicate its mass in kilodaltons. Now, GP350 resembles C3dG, so that when the B cell detects the EBV approaching, it registers that GP350 molecule as C3dG, and so it accepts it. It thinks that nothing's wrong, surely. It's just a regular pathogen. It's not. And now, at this point, EBV's goal is to enter into the B cell, and it does this by fusion. Now, GHGL and GP42 are used to initiate fusion, but GB is used in fusion itself. Now, when it infects a B cell, it causes them to multiply, creating lymphoblastoid cell lines, LCLs, where an EBV establishes latency, and that's its main virulence factor, is that it can remain latent for years and years and years. So, and it targets B cells in the peripheral blood, that is, the blood that is currently in circulation. But it infects more than just B cells, as I've said before. It infects oropharyngeal epithelial cells, but more specifically it attacks the tonsils and adenoids, and the reason it does this is because they also express CR2. Now, it replicates lytically in epithelial cells. In B cells, EBV typically remains in a latent state, and when its genome enters the B cell, it's not integrated into the B cell genome. And in these cells, it replicates as an episome, which is copied every time the B cell divides. Otherwise, it remains in a latent state, except for individuals with weakened immune systems, such as older people, in whom EBV may reactivate and again cause symptoms of mono. However, even as with the initial infection, this doesn't always cause symptoms. How is it transmitted? It's typically transmitted through bodily fluids, most often saliva, but it can also spread through sexual contact or blood, such as in organ transplants or blood transfusions. EBV is shed continuously into the saliva, like I said before, and that replenishes the regular virus level every time the infected host swallows, which is about once every 2 minutes under regular circumstances. So it can be transmitted by blood by those infected B cells, because again, B cells, it infects the B cells in the peripheral blood, the blood that's in circulation. So, or it could be infected, it could spread by saliva, because your body is shedding oropharyngeal cells into the saliva. It's typically transmitted by human carriers, but it can also be transmitted through non-living reservoirs, such as toothbrushes or utensils, supposing that there is saliva on those objects, such as in this toothbrush right here. If there's fresh saliva on there, and that saliva contains epithelial cells which are infected with EBV, then it can pass on to someone else, if for whatever reason you're sharing toothbrushes. Don't do that. How do you get tested for mono or EBV? So in order to determine whether an individual is infected with EBV, there are 2 blood tests that may be conducted. And you use blood tests because, again, it infects B cells in circulation. That just makes sense. So EBV in both its latent or active states consistently resides in the B cells within the peripheral blood. And these blood tests check for certain antibodies within the host. And these can arise during and remain for quite a while. I've heard it remains for years afterwards, after an EBV infection. It may even remain for your whole life. So these, particularly you're checking for anti-VCA IgG, VCA standing for viral capsid antigen. It typically takes 6 to 8 weeks after an infection to form these antibodies. So when you get tested, when you get a blood test for EBV or for mono, they may have you come back a week after, even if you test negative, because although you may experience symptoms of mono, it takes a few weeks to form those antibodies. Another way to judge whether you have mono is by lymphocyte count. An increase in lymphocytes may denote that you are infected with EBV, and a typical lymphocyte count, it's like 1,000 to 4,800 micrograms per liter. Neutrophil count, if it decreases, then you could also be infected with EBV. And the two blood tests that may be conducted are monospot and EBV tests. Monospot checks for heterophile antibodies, and the EBV antibodies tests checks for other antibodies. Now, EBV in the blood is a lifelong infection. Is there a cure? Sadly, current research is quite conclusive here. There is no cure or vaccine for EBV. The reason that I earlier detailed GP350 and its role in EBV attachment to the B cell is partly because it's interesting, but it's really because it really is important. That's where research has been focused on for the past few years. It's been focusing on the GP350 because if you can prevent attachment of EBV to the B cell, then you prevent infection. Problem solved. But possible future research may include research into AMO1, which is apparently it blocks entry into the B cell by EBV. It has something to do with blocking GHGL. And there are also mouse antibodies that are being looked into. So anyway, that is my presentation on mononucleosis. I hope you enjoyed it. Thank you for watching.